Okay, hello everyone. Welcome to our latest installment of the Bioinformatics Lunch and Learn webinar series at PacBio, where today I will be talking about our new microbial assembly pipeline in SmartLink version 8. So our agenda for the day, we'll start with some introduction and background information. We'll go into the algorithm under the hood and how things are working. Talk a little bit about the graphical user interface and how to use that to complete this analysis. Go through some of the results that you will see in the graphical user interface. Then do a command line example, and finally close it out with a brief overview of some useful resources. So my name is Dan Browne. I am a bioinformatics field application scientist for PacBio. I'm based out of Chicago. On the line with me today helping out is Matt Seaton, my colleague who is based out of San Francisco. There are two other BFX FAS, as we call ourselves, Kay Weber and Carmen Gorko. And I've just included our emails here, so if you ever wanted to reach out to your nearest Bioinformatics FAS, feel free to do so. So today, of course, we'll be talking about microbial genomics. And I wanted to start off with this overview of microbial genome complexity and highlight the importance of having long reads for microbial genome assembly. This figure on the right here shows really nicely that PacBio long reads make a very significant difference in the quality of the genome assembly. You can see the CONTIG N50s for the PacBio assemblies are much higher than those done with the MiSeq data, and additionally the number of CONTIGs assembled with PacBio is much lower, approaching the number of chromosomes. So for the purposes of assembling these microbial genomes, it's really important to get these long reads that can span repetitive elements that would otherwise obfuscate the assembly. So next slide here. So this is just a brief overview of our microbial barcoding and sequencing pipeline that we've developed, starting from gDNA, going through shearing, library construction, barcoding and pooling, optionally size selecting, and finally getting it onto the sequencer itself. And then what we'll talk about for the rest of this presentation is analysis with SmartLink. But the rest of the first 5 steps are typically doable within a day or two. So this protocol has gotten pretty well optimized and can be done reasonably quickly. What about cost? So with the Sequel II, this has made a very large difference, and our best estimate, you can see at the bottom, getting straight to the point, is that for a 48-plex of microbes, it'll cost you about $70 a microbe, and that'll get you a high-quality assembly for each and every one of the microbes in that 48-plex. And I believe this can accommodate up to 300 megabases of total genome sizes. So that is to say that the 48-plex should total, all the genomes should add up to, I believe, 300 megabases or less. So to prove that, and to show you that we didn't just make that up out of nowhere, we demonstrated this by taking 15 bacterial strains, preparing 48 libraries. You can see that a number of the strains were done in triplicate. And then we multiplexed these 48 libraries from these 15 strains and sequenced them on the Sequel II. You can see the expected genome sizes there range from 1.7 megabases at the low end up to 8.4 megabases at the high end. Also of note is that that one strain, Burkholderia cepacia, has 3 circular chromosomes, whereas the rest of the strains just have a single circular chromosome. You can also see on the far right column the plasmids, the known plasmids that we expect to recover from this pipeline. So, highlighting the sequencing data that was generated with this library, we got a total of 104 gigabases of polymerase read bases from 2.7 million polymerase reads, with an N50 polymerase read length of 87 kb. The total unique molecular yield was 28.9 gigabases. So that's essentially the sum of the length of the templates in each of the ZMWs on the chip. Another really nice plot that we produce that summarizes the data well is this one here on the left that you see, showing the insert read length by the HQ read length. That's essentially the insert length by the polymerase read length on the x-axis. And so what you can see is that there's this hot spot here between 10 and 20 kb on the insert read length axis. That tells you that the fragment sizes are most highly concentrated between 10 and 20 kb. And you can see that we've made many passes on these, getting up to 20 kb, up to 50 kb and above on the total polymerase read length. So that tells us that we have multiple passes over these 10 kb inserts, which is a nice thing. So after demultiplexing the barcodes from this library, you can see the distribution of polymerase reads per barcode here. And there's some variance, which is to be expected, but there's a pretty good coverage across the range. And we'll show some results from Assembly very shortly. I would just like to point out that this data set will be available for download very soon from our DevNet page, which is linked through GitHub. You can follow that link. These slides will, of course, be sent out following this talk. And so you'll be able to click that link, or you can probably just find it through Google as well. It's not up there right now, but it will be up there very soon. So keep an eye out if you're interested. All right, back to the assembly. Results here, you can see all 48 of the assemblies yielded complete chromosomal contigs. For the Burkholderia species, you can see there's 3 contigs, but again, that is because there are 3 chromosomal contigs in that species. The rest of them all perfectly assembled to 1 contig. What about the quality of those contigs? So to assess that, we compared our assemblies against some reference genomes that were available and calculated the average quality value for each of the species. We actually found that a couple of the references didn't seem to match, so we think there was some mislabeling, and those have been excluded. So if you're looking at this in more detail after the fact, you might notice that there are not all of the strains here. That is why. But for the ones there that the reference matches what we were expecting, we compare our assemblies against those references, and you can see that the QVs are all well above QV30. That means 99.9% accurate for QV30. QV40 would be 99.99% accurate, and so on. So our assemblies are pretty accurate and giving good results, all the way up to just above QV60 for that one species in particular. What about plasmids? So one feature that I'll explain in more detail soon is the plasmid assembly step. But essentially, in this pipeline we'll assemble chromosomal contigs, and then also we'll assemble separately the plasmids. So you can recover those as well. You can see that of all the strains that we're expecting plasmids, the majority are recovered as complete plasmids. That at the top there, you'll see that in the Bacillus cereus strain that 2 of the 3 assemblies, we did not recover that plasmid. But I just want to point out that's a bit of an edge case because that plasmid is actually a linear plasmid, not a circular plasmid. So it behaves a little differently as the algorithm was really designed to assemble circular plasmids. And it works well for everything else you can see. I would also just like to point out that in the Klebsiella pneumoniae species, the 2 kb plasmid is missing. But that's not necessarily a function of the assembly algorithm, that's more a function of the library preparation. Because if you do a size selection on your library and remove all pieces of DNA smaller than, say, 5 kb, you are going to throw out any plasmids that are smaller than 5 kb, and so you might miss that. So if you're looking for small plasmids, just be conscious of the fact that if you do a size selection, you may lose out on some of those smaller reads. Okay, so how is this working? What's going on under the hood here? Our traditional assembly process that you may be familiar with is the hierarchical genome assembly process, or HGAP. Our latest release now has been HGAP4, and that has traditionally worked pretty well for assembling a wide range of genome sizes. However, we have also noticed that it doesn't always perfectly assemble these small circular chromosomes with the default parameters. So we thought, well, maybe we can build something better and also add in some new features. So we built Microbial Assembly, which is intended to assemble small prokaryotic genomes, and we're really focusing HGAP4 now for larger eukaryotic genomes. But while we were at it, building this new pipeline, we decided to add in some new features. So here's a high-level overview of the whole assembly pipeline. You can see it starts from the raw reads to a chromosomal assembly step that assembles the large circular chromosomal contigs. There's then a mapping and filtering step that recovers reads that do not map to those large contigs. And then all of those reads are reassembled in the plasmid assembly stage that then assembles those plasmids using tuned parameters to do so. That is followed by a mapping step, polishing, an origin of replication rotation, and automatic preparation for NCBI. So I'll highlight that origin of replication rotation a little bit later on, but that is another one of the nice new features of this pipeline that orients the assemblies around the estimated origin of replication. So looking at the assembly step, what's different between HGAP4 and microbial assembly? One big change is that we have taken out the D-aligner tool stack, which was developed by Gene Myers, and replaced it with this new tool called Raptor, which is a graph-based mapping and alignment tool. We also added in a new chimeric read detection method, which really helps to improve assembly results as well. However, we are reusing the string graph and error correction components from the HGAP pipeline, which is part of the Falcon package, for those who are familiar with that. But you can still see it does essentially follow the HGAP paradigm of overlapping raw reads against seed reads, error correcting those seed reads, overlapping the corrected pre-assembled reads, or P-reads as we call them, and then generating the assembly from that. So this chimeric read detection method is really nice for cleaning up the chimeric reads that might be in the read set. It inspects overlaps from the P reads and constructs a graph like this. And here visually you can see pretty clearly that this is like a chimeric read. So now these are being caught and discarded so that they do not interfere with the assembly. Raptor is very advantageous because it is a graph-based mapping tool, meaning that it doesn't just take a FASTA file and then align against that. It actually takes what is called the GFA file, or graphical fragment assembly, which has the linear contig information, but also contains information about the assembly graph. So, for example, in a circular assembly, your linear contig in the FASTA file is actually connected by an edge where the end of the contig connects back to the beginning, but that's not reflected in a FASTA file. So using the GFA, we can then provide that extra layer of information to the mapping tool, and Raptor is able to thread reads through complex regions, like you can see at the bottom where there's a bubble in the assembly graph, and Raptor is able to correctly map the read through that region and maintain awareness of that complex graphical structure. This is particularly important for circular assemblies. So PBMM2 is our canonical read mapping tool that is a wrapper around Hengley's Minimap2 tool. And you can see that tool, which is not a graph-based tool, loses coverage at the end of the contig because reads that would wrap around can't be mapped properly. Whereas Raptor at the bottom is able to thread the read from the end of the contig back to the beginning through that through the graphical information, and is able to maintain full coverage across the entire contig. This is really useful when it comes to polishing, because if you don't have proper coverage at the end of the contigs in the polishing step, you might get bad calls at the ends of the contig. Polishing is done using our Arrow model, which is a hidden Markov model implemented in our latest GCPP package, which you can utilize via SmartLink or via PBBioconda, for those who are familiar with that. And then the origin of replication orientation is done. This uses a GC skew calculation. This figure comes from the paper where this was demonstrated, and the reference is provided there at the bottom. So if you want to go read more about that later on, you can follow that reference there. But you can see that the minimum and the maximum of the GC skew indicate the terminus and the origin of replication. So this calculation is done in our pipeline. The minimum is determined, and then the contig is oriented such that in the FASTA file that is output, the origin of replication is at the beginning and the terminus is at the end. So this is useful because if you want to, say, align multiple genome sequences after you have assembled them, you want them to all be oriented in the same way so that they line up as you would expect. Finally, our pipeline produces a formatted file for submission to NCBI. So there's a number of guidelines, as you can see here, from the NCBI website, and we want to make your lives as easy as possible for submitting these sequences to NCBI, so we produce a file that is formatted for that, ready to go. Okay, so now we've seen under the hood a little bit. I just want to give a brief walkthrough of the GUI. Starting here, this is our SmartLink homepage. This is what you'll see when you log in. And if you follow this into the Smart Analysis portal, you'll see this Create New Analysis button, which you can select. Then you will want to enter your analysis name and search for your data. In this example here, you can see that I have found a demultiplexed data set that contained 10 barcodes. I've given the analysis a name, and I'll just go ahead and click on that little link there at the number 10, and that takes you to the demultiplexed data, where you can select whichever barcode you want to assemble. So we've selected one there, and we can click next, and this will bring us to the analysis application selection page, where you can click this drop-down menu and select microbial assembly. It gives you a few options. You can see you need to enter an estimated genome length, and then you can also select advanced parameters. There's just a few things that you can look at here. One would be the seed length cutoff. The -1 indicates an automatic seed length cutoff selection based on the seed coverage in that middle box there, which by default is at 30x, which is our recommended coverage. And then there's also this advanced options box where you can put in a number of key equals value pairs, if you so desire, to tweak some of the assembly pipeline parameters. We don't recommend doing this to begin with. The parameters have been pretty well tuned, and as you can see in the beginning, I was able to assemble all 48 of those demultiplexed barcode data sets into the one contig. But I just wanted to point this out. If you do want to make changes, these are the parameters that you can alter, and now you have this for reference. So once you've entered all of your options, you just click Start, and it is good to go. So once that has finished, you will see a page like this one that contains all of the analysis results. So the thumbnails has all of the plots that were generated. You can look at the polished assembly statistics. There's some summary metrics right here. You can see this assembly yielded 3 polished contigs with a maximum contig length of 2.9 megabases. This tab shows each of the polished contigs that was generated. You can see there's a chromosomal contig, that's the 2.9 megabase one, and it is circular, as well as a plasmid contig of 32 kb that is also circular. Occasionally, you will see artifactual contigs, like the one at the bottom there, that 4.3 kb contig, not circular. Sometimes there are artifactual contigs that come through the assembly process, but they're pretty easy to identify. Typically, they're not circular, they have weird coverage plots, and then if you blast them against NCBI, they will not yield anything significant. So you can also look at the alignment to the draft assembly. This is just prior to the polishing, and you can see that 97% of the bases aligned back to the draft assembly with a mean concordance of 90%, alignment length N50, 8 kb. But all of those metrics, I think, are also very nicely encapsulated visually in this plot here, alignment concordance versus read length. And so you can see on the y-axis is the map concordance, on the x-axis is the alignment length, and then on the z-axis is the count of the number of reads in each one of those XY buckets. And so what this graph is showing is that we've got a really nice concentration of alignments that are just about 90% or just above that of map concordance, ranging from all the way up to almost 15 kb in length of the alignment. So pretty good alignments, I'd say. You could also look at coverage plots. So this page here will show you the coverage plot for each of the contigs that was assembled. So here you're looking at the coverage across that large 3 megabase chromosomal contig, and it shows nice variance from about 600 to 400 or so, with a few peaks above and below that. You can also see the reflected here, the orientation of the origin of replication. Typically, the maximum coverage will be found near the origin of replication, and the minimum will be found near the terminus. So you can see that the orientation step did a pretty nice job. That's reflected in the coverage across the reference. Finally, you can find all of the, or excuse me, not all, but the key data files here are exposed in the GUI and available for download, so you can access the data, download it to your computer, and inspect it more closely afterwards. All right, so that was the GUI. Now I'll just give a quick rundown of the command line, how you can do this using our PBCROMWELL tool, which is part of our Smart Link or Smart Tools package. If you don't want to install the GUI, you can do a Smart Tools only installation, where you will still have access to the PBCROMWELL command line tool to run all of our pipelines via command line. So you can see here this PBCROMWELL show workflow details of the PB assembly microbial pipeline, and generate an inputs JSON file that I've named here as params.json. So when you run that command, you'll see this standard out shown here, the pipeline summary, task options. Once you've executed that command and generated the params.json file, you would want to edit the parameters accordingly in the JSON file to your specifications. Then you will need to generate a PBCROMWELL configuration file using the second command shown there, PBCROMWELL configure, with the default backend parameter. We've set that to SGE, but we also support other backends, Slurm, LSF, PBS, and then you specify the output file named here cromwell.conf. And then to execute the pipeline, you see the last command there, PBCROMWELL run, PB assembly microbial, with the -E option stands for entry point. That points to the subread set that you want to assemble. You specify the full path to the Cromwell configuration file, the params.json file, optionally the number of processes that you want to execute, and any queue that you want to submit your job to. This is the params.json file for the microbial assembly pipeline. On the command line, you can see that a lot of you can specify the number of processes there. You can also specify entry points for subreads, or you can do that via the command line task options. You can also edit the advanced options similar to how you would in the GUI by specifying those key equals value pairs in the microasm advanced options flag right there. As well, you can edit the coverage. Most importantly, the genome size needs to reflect your estimated genome size. And that's it. So again, not a ton of stuff to edit here, which is good. You know, we want to keep it simple for you, but you can, of course, always modulate those advanced options if you feel like testing that out. Once you finish running Cromwell, the PBCROMWELL pipeline, you'll see an output folder like this that contains the Cromwell configuration file, a directory of all of the executions, all the steps in the pipeline are contained in that Cromwell executions directory. Your inputs JSON, a logs directory, metadata, options, the outputs, which is pretty important. That's one you will most commonly use. The workflow description language file that describes the actual pipeline Cromwell ran, and then a running shell script. So if you look into the CHROMOSOMES executions directory, you'll see nested directory structures like this. So each of those directories contains further subdirectories that have all of the scripts, inputs, outputs, everything for each step in the pipeline. So if you really wanted to get into the nitty-gritty and look at the data from each step in the pipeline, this is where you will find it. The outputs folder is intended to just give you some pointers to the key output files from the pipeline, and so you don't have to go digging through all of those nested subdirectories in the executions. This provides symbolic links to each of the key files that you would probably want to access. So that is just about it. We'll wrap it up here with useful resources and then address any questions that came up during the presentation. I just wanted to point you to, we have documentation of procedures for all of these things on our website. You can see we have a few different pieces of documentation. Again, all available through our website. So if you're interested in reading more about like the library preparation or loading the instrument, anything with respect to the workflow, you can find that in these documents on our website. Also, I'd like to give a shout out to any SQL users who might be listening. We have a new version of the instrument control software coming out very soon. I think Q1 next year. That will give you some really nice improvements, fix some known issues, and also add a new feature which includes automatic detection and recovery of inserts with missing adapters, which is really important because those can be a problem sometimes. So if you're a SQL user, I'd highly recommend keep your eyes out for ICS version 8. It's coming soon. Also, I just wanted to point to our tech support team. We have a fantastic tech support team who's available to answer your questions throughout the day. So if you have any issues that you've run into or anything like that, don't hesitate to reach out. We love hearing from our customers, and we're really happy to help. Finally, we will be having our next Bioinformatics Lunch and Learn webinar in Q1 2020 sometime. So keep your eyes out for that. We'll be announcing a date probably early in the new year. We will also be at PAG in San Diego from January 11 to 15. So if you'll be there for that, come say hi. We'd love to chat. If you have any questions or comments or suggestions for our next topic, feel free to send me an email. That's dbrowne@pacb.com. You will also have the opportunity to provide feedback anonymously in a follow-up survey that'll go out after this is over with a copy of the slides and a little survey. So, if you don't want to email me directly, you could also provide feedback or make suggestions through there as well. So, with that, thank you so much for listening. I think we have a few questions. If you haven't entered a question yet, you can do so through the dialog box, and we will take a look at those now and see if we can answer. Let's see. I think, how do I read this? Oh, okay. So, got one question, said, I've seen a few genomes assembled using HGAP3 and HGAP4. Does this pipeline work for RS2 data? Yes, yeah, it will work with RS2 data, I believe. So you should be able to go ahead and use it with any older data, generating newer assemblies. But if you do hit any issues with that, again, feel free to reach out to tech support, and we'll help you work through that. Oh, I'm sorry. I think I actually am wrong there. My apologies. One of my colleagues is telling me that SmartLink 8 only supports SQL data. So I'm incorrect. Please ignore my comment there. SmartLink 8 will not work with RS2 data. My apologies. Okay, let's see. Is the Slurm CROMWELL version support assembly on multiple nodes simultaneously? If I understand that question correctly, it is asking whether CROMWELL can make use of multiple nodes to perform the assembly. And if that is the question, as I understand it, yes. CROMWELL was designed by the Broad Institute and enables the efficient scaling of computation across a computer cluster. So that's why we have implemented CROMWELL now as the backend in SmartLink 8, because it's really good and it scales really well across clusters and makes support a lot easier. And yes, Slurm is supported. Okay. Are there any other questions or comments? Okay, I'm not seeing anything come in, so I think we can call it a wrap there. Thank you again, everyone, for your attention and participation today. And again, if you have any questions or follow-up stuff, feel free to reach out to us via email. With that, thank you again for your attention and have a good day.